Hello and welcome to this PowerPoint where we're going to look at the different factors that affect photosynthesis. The specific points from the study design we'll be focusing on, as you can see here, the general factors that impact on enzyme function in relation to photosynthesis and cellular respiration, such as changes in temperature, pH, concentration, competitive and non-competitive enzyme inhibitors, and also the factors that affect the rate of photosynthesis, light availability, water availability, temperature, and carbon dioxide concentration. Now, that first point did also mention cellular respiration. We won't be focusing on cellular respiration in this PowerPoint. That will be in a later PowerPoint. So this will only be focused on factors affecting photosynthesis. Now, lots of different things can have an impact on how quickly photosynthesis can happen and whether or not it's able to happen at all. There's sort of four categories I've grouped these into. You can think of them as substrate concentration factors, temperature, pH, and inhibitor factors. And so under substrate concentration, that could have to do with carbon dioxide concentration, the availability of light and its intensity, or even water availability can have an impact. So those are all the sorts of factors that can actually play a role in impacting whether or not photosynthesis even happens, and then also how quickly it can happen. The other thing that can have an impact on photosynthesis is what kind of plant are we actually talking about? Is this a C3 plant or is it actually a C4 or CAM plant? And sometimes the different types of plants actually respond, or their ability to do photosynthesis is different depending on some of these different factors. And so we'll look at how each of these plants respond to when we change these factors, and we'll have a look to see, do they respond in the same way or do the plants actually have slightly different responses? So the first thing is substrate concentration. And so generally speaking, you need to remember that with any chemical reaction, there is always going to be an impact that the amount of a reactant or a substrate will have on how quickly that reaction happens. And generally speaking, with any reaction, specifically those mediated by enzymes, you're going to end up with rates of reaction graphs that look very much like the one on the right there. We've got a couple there. The first one's very basic. It's just showing you that as we increase substrate concentration, the rate of reaction increases to a point. And so this is a pretty classic shape where it increases really quite rapidly and then it plateaus and the rate of reaction doesn't get any faster. This doesn't mean that the reaction has stopped, it just means the reaction is not going any faster than it has been going. So that's what that graph means. If you look at the little one beneath it, you'll see with enzymes why this might actually be. As we increase the substrate, those enzymes will actually get to a point where they're overwhelmed. So they're completely in use, they can't go any faster because there's not any more free enzymes. And so it doesn't matter if you keep increasing the amount of substrate you have, that reaction can't get any faster because there's just not any more enzymes to facilitate that reaction. So that's part of why it plateaus. It's going as fast as it possibly can and it just can't go any faster. Now with photosynthesis, there are two reactants or substrates, and that is carbon dioxide and water. But with photosynthesis, as we know, light is actually essential as well. It's not actually a reactant, but it's required for photosynthesis to occur. So light can, in a way, act a little bit like a substrate in terms of how it can affect the rate of photosynthesis. And we'll look at each of these in turn. So first of all, carbon dioxide. Pretty simply, as carbon dioxide, as its concentration increases, generally the rate of photosynthesis increases. As carbon dioxide's concentration decreases, the rate of photosynthesis decreases. It's nothing revolutionary. It's pretty straightforward. The more of the reactant we have in terms of carbon dioxide, the more that whole photosynthetic reactions can occur. So overall rate of photosynthesis is faster. But obviously there's going to come a point where you've maxed everything out and it can't go any faster. And that's what that plateauing point is. What that means is that before you reach that plateau, the amount of carbon dioxide is actually limiting how quickly this reaction can happen. So the carbon dioxide is acting as a limiting factor. But once we hit that plateau, what we're seeing is that we've actually got plenty of carbon dioxide. There's something else that's limiting how quickly that can happen. That might be another reactant, it might be the enzymes available, it might be the energy available. There's quite a number of things that could be limiting that reaction aside from carbon dioxide. So that's one way you can actually read those graphs, is you can see the point at which the thing you've been increasing is no longer actually limiting the reaction and it's going as fast as it can because something else is now limiting it. Generally speaking, this is often down to all the enzymes are being utilized, and the reaction just can't go any faster because the enzymes that would be required are fully saturated. Now, one thing to keep in mind with photosynthesis is that we know when carbon dioxide levels get too low, it can actually lead to photorespiration. So carbon dioxide increasing, rate of photosynthesis increasing. That's nice and easy. But you need to remember, not only when carbon dioxide decreases does the rate of photosynthesis increase, you also run the risk of increasing photorespiration, which is a completely different sort of process and a really wasteful one at that. So that's how the levels of carbon dioxide can actually affect photosynthesis. So how then does this affect or differ between C3, C4 and CAN plants? This is actually one of the factors that you'll see the biggest difference in how they deal with this. So as we were saying, if those carbon dioxide levels get too low, it can lead to photorespiration. So this is where our CO2 levels got really low and the oxygen levels have gotten really high. So much so that the oxygen's binding to the rubisco instead of the carbon dioxide. Now, if you're a C3 plant, you're stuck. You have no specific mechanism for dealing with this, so you will, unfortunately, get stuck doing photorespiration. C4 plants and CAN plants actually have their own strategies. Remember, C4 plants put the light independent reaction in 2 different cells or spread it over 2 different cells, whereas CAN plants will actually do it at 2 different times of the day. That's how they split it. Those are their strategies to help reduce the chance of this CO2-O2 imbalance. And so that's how they work to reduce the chance of them doing photorespiration. Now, if you look on the graph, you'll see a little bit that's been circled. This is where we're looking at photosynthesis almost looks like it's going backwards, but the rate of photosynthesis is now below 0. And what we're talking about here, this is where the carbon dioxide level is so low that for the C3 plant, it's actually doing photorespiration and it's making carbon dioxide. So this is actually a problem for it. It's doing the wrong thing. You'll notice that the C4 plant never drops down here, and that's because of the mechanisms in terms of separating between the mesophyll cell and the bundle sheath cell that the C4 plant does. So you can see there that because of its adaptations, the C4 plant never starts doing photorespiration even when those carbon dioxide levels are low, because it has a specific mechanism for dealing with that. You may notice at the other end, the top end of the graph, where the carbon dioxide levels have gotten really high, that the C3 plant suddenly gets above the C4 plant, and that's just because at the end of the day, because C3 plants don't have any specific mechanisms and they do the most basic form of photosynthesis, it's actually the most efficient form. It's just really bad for them when the carbon dioxide levels are low, and so that's where their negative is. But actually, when levels are high, it's really quite good. C4 plants are still reasonably efficient. They're just not as efficient because they have that extra step. So that's why they sort of plateau a little bit earlier before C3 plants, and they don't quite have as high a rate of photosynthesis. It's not a major difference, but it's a little bit of a difference, and that's why you see that change at the top end of the graph. But for the most plant, you'll find between C3, C4 and CAN plants that there isn't going to be much of a difference when there's high levels of carbon dioxide. It really comes down to what happens when you've got low levels of carbon dioxide. So that is how carbon dioxide is going to affect the rate of photosynthesis. The next thing to talk about is light. And you might notice that the graph for how light intensity is affecting the rate of reaction is very similar to how carbon dioxide affected it. And that's because it's acting like a reactant. And this is a very classic shape of a graph for how changing your reactant or substrate concentration will affect the rate of reaction. So unsurprisingly, as the amount of light increases, the intensity of light increases, so does the rate of photosynthesis, and as the amount of light decreases, so too does the rate of photosynthesis decrease. Now, again, you've got a point at which it plateaus, and that's because everything within the reaction, everything else that's involved is being fully utilized, and so the reaction can't go any faster. It could also be you might keep increasing the amount of light, but there isn't heaps of water, and so that ends up limiting it. There can be other things that limit it, but generally it tends to be the enzymes. Now, it is worth noting that with light can affect photosynthesis in 2 different ways. You can talk about the amount of light in terms of the intensity and how much is actually available, but you also need to think about almost like the quality of the light and what wavelengths are actually present. And this goes back to the fact that plants use pigment molecules to help them absorb different wavelengths of light. Now, most plants use chlorophyll, to help them there. And with chlorophyll, what it likes, it really likes the blue end of the spectrum and the red end of the spectrum. It will absorb those wavelengths of light quite a lot. Green, it doesn't absorb. In fact, it reflects green light, which is why we see plants that use chlorophyll as green, because they're reflecting green light. So if you want to grow a plant, plants are outside and they're using chlorophyll, they're getting hit with sunlight. And for the most part, sunlight is what we'd consider white light. And so it actually contains the whole spectrum of those colors there within that. When you actually, it's a bit odd, when you combine all colors of the rainbow, you actually get white light. When you split white light, that's when you see a rainbow, and that's actually how rainbows are made. It's when the white light from sunlight hits a raindrop and is split like it's going through a prism, that you suddenly see all these colors of light separated into a rainbow. Now, all that, when it's combined, is white light, and if that's hitting the plant, that's great. It'll absorb what it needs, reflect what it doesn't. If you're trying to grow a plant inside and actually use artificial light to grow it, you would want to make sure that you're not just shining green light on the plant, because it's not going to absorb that. In fact, you could actually skip shining green light on it altogether and just shine blue and red light on the plant, because that's what it's going to absorb. So that's what's actually needed to provide energy for photosynthesis. So it's worth remembering that with light, it's not just how much light that's going to affect photosynthesis, it is the color or the wavelength of light that's also going to have an impact. That graph there is just showing you that obviously if chlorophyll is absorbing at the blue end and the red ends of the spectrum, then those ends of the spectrum is where you're going to have a lot of photosynthesis able to happen, because it's absorbing a lot of it. And so you see these peaks of rates of photosynthesis over the blue end and the red end, but it's really low over the green, and that's because it doesn't absorb the green, it's reflecting green light. And so the rate of photosynthesis, if you were only showing green light, would be very low, because it's going to reflect most of that light. So it's worth keeping in mind the two different ways light can affect photosynthesis. So how does this impact C3, C4 and CAN plants? Well, so it's important to remember that the light is important for the light-dependent reaction in photosynthesis. And for C3, C4 and CAN plants, the light-dependent reaction is exactly the same. So changing any part of the light is going to affect them all in the same way, because they all use it the same in their light-dependent reaction. Where they're different is in their light-independent reactions. So because they all have that same light-dependent reaction, they're all affected in the same way when you change the amount and the color of light that you're exposing to. Now, one important point I've got down the bottom there is if you're talking about an increase of light intensity, say outside when it's hot, it's a hot and sunny day, and you know that sunniness is more than normal or something, you're also talking potentially an increase in temperature. So sometimes, particularly out in nature, when light intensity is increasing, we also see an increase in temperature. Now, if that is happening, then yes, that can have an impact on water balance, that can affect carbon dioxide levels, and the whole light independent thing, and yes, you'll get a different response for them. But that's not so much because the light is different. That's because the change in the light is occurring alongside a change in a different factor, and it's that other factor that's having the impact. So it's just keep that in mind, that if everything's controlled, changing the light won't have an impact, won't impact the plants differently. If you're not also controlling temperature, and say it's a hot environment, becomes hotter and hotter as you increase light intensity, then yes, you may also see a difference, but it's not actually because of the light as much as it's because of the temperature. And so that's something to keep in mind. All right, this brings us to our first exam question. So it says, an experiment was carried out at a constant temperature and with a constant carbon dioxide concentration in order to determine the effect of changing light intensity on the photosynthetic rate. The following is a graph of the results. Based on your knowledge and the information in the graph, which one of the following conclusions can be reached? A. Photosynthesis ceases to occur at a light intensity of 14 arbitrary units. B. Plants do not undergo photosynthesis at a light intensity of 1 arbitrary unit. C. Light intensity is a limiting factor when the photosynthetic rate is less than 40 arbitrary units. And D. Increasing the amount of carbon dioxide at light intensity of 16 arbitrary units would lead to a decrease in the photosynthetic rate. So take a moment, pause the video and go and work out your answer, and you can then go and check your answer by watching the answer video linked in the description box below. All right, let's also now talk about exam question 2. So you have a bit of information that's going to be used to answer the 2 questions on this page. It says the graph below shows the net output of oxygen in spinach leaves as the light intensity is increased. Temperature is kept constant during the experiment. Question 1. Which one of the following conclusions can be made based on the graph? A. At point T, photosynthesis is no longer occurring. B. The optimal level of light intensity for photosynthesis is 40 AU. C. At point S, the amount of oxygen output is 1/3 of that at point P. Or D. Below 10 AU of light intensity, the aerobic respiration rate is greater than the photosynthetic rate. So that's the first question. The second one says the rate of oxygen output remains constant between points P and O because A. Heat has denatured the enzymes involved in the photosynthesis reactions. B. The concentration of available carbon dioxide limits the rate of photosynthesis. C. The light intensity has damaged the chlorophyll molecules present in spinach chloroplasts. Or D. High levels of oxygen produced at point P have accumulated around the spinach leaves, resulting in no more oxygen being produced. So you can pause the video, have a bit of think, answer those 2 questions, and once you've got the answer, go and watch the answer video for this linked in the description box below. Alright, let's talk about water and how water affects photosynthesis, because it's quite an interesting one. Really, the availability of water has an impact in a couple of different ways on photosynthesis. So obviously water is required for the light dependent reaction, so it's actually a reactant that's required for photosynthesis. So if you don't have enough water available, the reaction's not going to go ahead, and obviously the rate of photosynthesis would drop. Now, this is actually probably less common, particularly in nature, as to how water is going to have an impact. And that's because plants, for the most part, tend to have a lot of water that they've got access to in the soil, so it's not so much an issue. Now, obviously, droughts or long periods of hot weather can reduce the water availability, and therefore it can have an impact. But plants do also have certain mechanisms to help avoid themselves becoming dehydrated and losing too much water. So they generally try to keep sufficient water within them so that they can do the reactions that they need to do. So it tends to be less likely that water is going to become, say, a limiting factor compared to some of the other factors. That doesn't mean it can't, it just is a little bit less likely. What does happen is that the amount of water a plant has an impact on whether the stomata are open or closed. So the stomata, as you should remember, they're opening in the leaves, they're little pores, little openings, and they're made by 2 guard cells, and the guard cells are next to each other. And when these guard cells have lots of water inside of them, because the plant's got lots of water, so they're able to have lots of water, then the guard cells are all rigid and they sort of push against each other. And as that happens, there's a little gap that's created between them as they're sort of pushing against each other. But when water is not in great availability, it's a bit limited, and the plant wants to conserve what it has, what will happen is the water in the guard cells is reduced. And so they're not as stiff and as rigid, and they become floppy and flaccid, and they'll flop back down next to each other. And that actually closes off that little gap that was between them. And so that's essentially what we talk about, the stomata being closed. That's what's happening. And so when water levels are low, these stomata will actually close, and that means no water actually leaves through them. And that's really good because it's able to hold on to the water, so it's not continuing to lose it. It's a bit like if, and this sort of does happen in humans, if we're really dehydrated and it's very hot, normally when it's hot we sweat, but when we start getting really dehydrated, our body has a process where it actually stops us sweating just to conserve that last little bit of water we have. Now, for humans, if you get to that point, you're in big trouble because that's like severely dehydrated and you need urgent medical help. But for plants, it's pretty normal for them, when they're a bit low on water, for their stomata, their guard cells, to become floppy and to flop back next to each other and shut the stomata, and so therefore conserve and stop them from losing any more water. Now, that then will have an impact on the carbon dioxide and the oxygen that normally move through those stomata as well, and we'll talk about them in a moment. But this is just what I'm talking about. You can see on the left there that when the guard cells have lots of water in them and they're really swollen, they sort of push against each other and create this gap between them, and that's the stomata. But when they've got less water, they don't have that pressure and the ability to push against each other like that, so they flop back towards each other, and that gap gets closed off, and so that's this closed stomata. Now, that's essentially what's happening, and that's really good for the plant in terms of keeping the water it has. Where it's not good is what it means is as soon as those stomata close, carbon dioxide cannot come in through it and the oxygen cannot go out through it. So inside the plant, what happens is you start having oxygen build up, and because the carbon dioxide is being used, it starts dropping. And this is going to lead to a decrease in photosynthesis over time, but it will also lead to an increase in photorespiration. So when there's low water, we actually have to start considering how this is going to have an impact on photosynthesis and what might happen in terms of photorespiration. So yes, water might limit the rate of photosynthesis in terms of the light-dependent reaction not being able to go ahead if there's not enough water, but what's more likely is that this is going to happen, that low levels of water are going to cause the stomata to close, and that's then going to have an impact on the carbon dioxide and the oxygen levels. And this is where we do see a difference between our C3, our C4, and our CAM plants. Obviously, if C3 plants are having this happen to them, then they're the most susceptible of starting photorespiration because they don't have any mechanism to deal with it. So if you have low water levels in a C3 plant, and if you decrease that, you would expect to see less and less photosynthesis and arguably more and more photorespiration. And that would be happening for the C3 plant. On the other hand, C4 and CAM plants, because they have their own specific adaptations, what it means is that as those water levels drop and the guard cells shut and subsequently the carbon dioxide levels decrease and the oxygen ones increase, you would expect the C4 and the CAM plants to reasonably be able to keep doing photosynthesis as well as they have been doing and not really change. It might decrease a little bit, but not to the amount a C3 plant would. Now, it is worth noting that doesn't mean they won't do photorespiration. If the water availability is extremely low, they do become susceptible to doing photorespiration. So it is possible for that to happen, but they're much better at resisting it when the water levels get low. So that is how these plants will respond differently with water levels. Now, that has got to do with low water levels affecting the stomata and subsequently the carbon dioxide and oxygen levels. In terms of just low water levels affecting reactions in photosynthesis, they would all be affected the same way because the water is used in the light-dependent reaction at the very beginning, which they all do the same. So in terms of that, they would all be affected the same way. It's only when you're talking water levels affecting whether or not stomata are open or closed that you'll see a difference between how they respond to that. So that is how plants deal with changes in what are essentially substrate concentrations. But there are other things that can happen, and this is where you need to remember that photosynthesis is being catalyzed. All these reactions are occurring because of the action of enzymes, and there's heaps and heaps of enzymes at play. You've got photostem 1 and 2, ATP synthase, rubisco, pep-carboxylase, carbonic anhydrase, malate dehydrogenase, PPDK. They're just some other enzymes that are involved in photosynthesis, and you do not need to remember all of them. Probably the only two in that list I'd say you need to know are ATP synthase and rubisco. And ATP synthase probably we talk about more in cellular respiration than we do in photosynthesis, but it's in both of them. So there's heaps and heaps of enzymes. And what that means is when something can affect an enzyme, generally speaking, it's going to have an impact on the reactions that enzyme catalyzes. So if there's something that can affect enzymes, that's also going to then affect photosynthesis. So obviously we've talked about substrate concentration, but some of the other factors that can affect enzymes are temperature, pH, and inhibitors. And so because temperature, pH, and enzyme inhibitors can all affect enzymes, it means, in this case, if we're talking about the photosynthetic enzymes, these can also affect the rate of photosynthesis. And we're going to look at how they each can do that. So the first is temperature. Now, when we looked at how substrate concentration affects the rate of reaction, what we saw pretty much consistently was as you increase the substrate, the amount of substrate, that rate of reaction increased until it couldn't increase anymore and it just was going at the top speed it could. With these other ones, you'll start noticing different shaped graphs. So here, as temperature is increasing, what we tend to see with the rate of reaction is that it increases and increases and increases. Now, if this was a reaction that didn't use enzymes, you'd probably just expect that to keep increasing. But because it uses enzymes, we actually get to this point where it's sort of as fast as it can go, and as soon as you increase the temperature past that point, it drops off really suddenly. So if you increase the temperature, generally you'll increase the rate of reaction, and if you decrease it, you decrease the rate of reaction. on a most basic level, the reason for that, for any reaction, has to do with the amount of energy the molecules have, and therefore how likely they are to collide with each other. So generally with chemical reactions, when you increase temperature, molecules get more kinetic energy and they move more. Therefore, they're more likely to collide and more likely to react. So we have an increase in the rate of reaction. Less temperature, or decreasing the temperature, means less energy available to those molecules. They move less, they're less likely to collide, or it's going to take them longer to collide. So we have a decrease in the rate of reaction. So just basic chemical reactions and speeding them up and slowing them down. So high temperatures equal high molecular energy, low temperatures equal low molecular energy. And so the higher the molecular energy, the more likely they'll collide and react. The lower, the less likely they'll collide and less likely they'll react. So that's the basics of chemical reactions. But because photosynthesis reactions, like a lot of other pathways in organisms, are catalyzed by enzymes, which are a type of protein, you need to remember how proteins get affected by temperature as well. So all that first part is true. Yes, as we increase temperature, molecular energy increases, things move around more, they're more likely to collide, rate of reaction increases to a point, because you've got proteins in the form of enzymes involved. And so there's going to get a temperature that once you pass that temperature, the proteins that make up those enzymes are going to denature. And therefore, once those become denatured, they stop functioning, they stop being able to facilitate this reaction. So it doesn't matter if you keep increasing the amount of temperature, the thing that is needed to do the reaction is no longer functioning. So we'll see a drop off in the rate of photosynthesis. So that's why we get this graph like we have on the right, where it's increasing, 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 gets to a point and then drops off. And that's because it's, yep, everything's moving quicker and quicker and quicker, or at our optimal point, and then denatured and not working. And that's what is on the far side. It's happened there. So it's really important when we're talking about reactions that are catalyzed by enzymes to remember that enzymes are proteins and they're affected by things that affect proteins, like temperature. What you want to know for a reaction is what is the optimum temperature, and that is the temperature at which that enzyme is going to be at its highest function. It's going to be working as quick as it possibly can, because at that point that's the sort of highest rate you're going to be able to get out of it. So what you'll find is as the enzymes approach their optimum temperature, overall rate will increase. The rate's approximately doubling for every 10 degrees increase in temperature. It's about that. Above the optimum temperature, the rate is going to decrease because the enzymes are denatured. And eventually, all the enzymes will, as you keep increasing the temperature, all the enzymes are denatured completely. And so you're going to have no function at all, and therefore no rate of reaction, as you can see in the diagram. Now, how does this affect our plants? Well, it's a bit of an interesting one, and we'll go through it. The plants are all obviously going to be affected by temperature in the same way, in the sense of that they all use enzymes. And so the temperature, as you increase, it's going to affect enzymes in the same way. It doesn't matter what they are. Now, you might find that, yes, some of them, because they are best suited for hotter environments, their specific enzymes may have a slightly higher optimal temperature than others. But at the end of the day, because they're all still using enzymes, you can expect them all to have that sort of graph where it increases to whatever their optimum is, and then you have a quick drop off on the other side as those enzymes become denatured. So generally, we would expect that same shape of a graph. Where it is in terms of temperature, it may be slightly offset depending on what they're best suited for. So they're going to produce graphs with the same general shape. Okay, don't worry too much about the diagram there. It's just a picture. It's a bit of a, potentially a bit of an inaccurate one. So just don't take what's on that diagram too much to heart. Now, what you sort of can expect in terms of who will do better and what temperature is C4 and CAN plants. Generally, they're better adapted to hotter environments, and C3 is more suited to cooler and milder environments. Now, the other thing to remember is obviously higher temperatures can make rubisco more likely to bind with oxygen, and that's true for all of them, which is why C4 and CAN plants have those adaptations that they do have. Okay, so you need to consider, okay, temperature is going to affect them all in the same way, in the sense of they all use enzymes, and therefore high temperatures will eventually cause all enzymes to denature. But where we're talking about them differently is how they deal with high temperatures in other ways. So we know that for all of them, a high temperature will start to make rubisco more likely to bind with oxygen. C3 plants have no way of dealing with this, but C4 plants and can plants do. Now, when temperatures are high, plants, particularly if it's dry as well and they don't have lots and lots of water, plants will close their stomata to help reduce that water loss. Now, that obviously starts to cause that imbalance of carbon dioxide to oxygen, and so C4 plants and can plants have two different ways of dealing with it. As we know, the C4 plants will basically separate where rubisco is from where the oxygen's being made and put them into two different cells, so that carbon dioxide has to be moved into a different cell so it can actually attach to rubisco. And can plants will actually do things at different times of the day to help build up a store at night of carbon dioxide, so during the day when they can't get new carbon dioxide, they can just pull out what they need from their store and give that to rubisco, so rubisco is not tempted to bind to oxygen instead. So that's sort of a second way that these plants deal with temperature differently, or changes in temperature will affect them differently. That you'll see that C4 and can plants will be able to compensate for that increase in temperature better, and will probably be able to, as temperature increases, probably still have higher levels of photosynthesis, whereas for C3 plants, it's going to start dropping off, not just necessarily because, say, rubisco's become denatured. In that bit of time before it denatures, it'll be because rubisco will start binding to oxygen and will start doing photorespiration in a C3 plant, whereas C4 plants and can plants can avoid that for a bit longer. Obviously, there'll come a point where the temperature is high enough that rubisco will just become completely denatured and it will stop working in all of them, regardless of where it is and what's around it. So there's sort of two ways to think about that. That is this sort of, as you just increase it, obviously at some point everything's going to get denatured and all fall to bits, but also then also consider how C3, C4 and can plants deal with hot temperatures where it's warm or hot, but not so much at the point where, say, all the enzymes have denatured. That's where you start to see a little bit of difference. Once you've gone above that, then it doesn't really matter. They're all denatured and it's not working anyway. So those are the two different ways those plants interact with temperature. This brings us to exam question 3, which says greenhouses have been used to generate higher crop yields than open field agriculture. To encourage plant growth in greenhouses, the conditions required for photosynthesis are controlled. Commercial greenhouses, like the ones shown below, often use a lot of energy for heating, ventilation, lighting, and water. First part, consider the reaction of photosynthesis. Why would it be important to maintain the temperature within narrow limits in a commercial greenhouse, and justify your answer? And then for B, scientists are developing a new material to cover greenhouses, which can split incoming light and convert the rays from green wavelengths into red wavelengths. Explain how this new material increases crop yields. Alright, so you can pause the video now and you can go and answer that, and then you'll be able to check your answer by watching the video linked in the description box below. So the next thing we want to talk about affecting photosynthesis is pH. So this is coming back again to photosynthesis catalyzed by enzymes, and enzymes are proteins. How does pH affect them? And so in a similar way to temperature, there is an optimal pH level, just like there's an optimal temperature. Extreme pHs will denature proteins. But with temperature, what we found is that low temperatures just meant lower rates of reaction until you increase the temperature until you reach your optimum, and then it's only once you're above your optimum do you start to denature it. With pH, it's not like that. You've got your optimal pH, and as you start to move either above or below it, if you get far enough away on either side, at some point you will denature it, either because it's in a too low pH or a too high pH. So we see this sort of bump with the pH graph, as you can see on the right there. That's generally the shape of the graph that we'd expect to see. It's more symmetrical than what we see with the temperature graph, where too low, protein's fully denatured. Too high, protein's fully denatured. Somewhere in between those points is your optimal pH. Now, with our 3 different types of plants, they're all going to be impacted by pH in the same way. They're all using enzymes. It doesn't matter on the type of plant they are. Now, you may find the enzymes they may use may have different optimal pHs depending on the environment they have to operate in. For the most part, you'd expect them to be all pretty much the same. And so, because they're using enzymes, anything too low or too high on the pH scale is still going to affect them in the same way. So you don't really see any difference between C3, C4 in canned plants, and they're all going to end up producing graphs that look like the one on the left there. There won't be any differences between them. So it is worth noting that pH for the most part just affects them all the same. Alright. Okay, so we're going to watch a little video just recapping some of these factors that affect photosynthesis.
We saw in the last video that plants need to carry out photosynthesis in order to survive. The rate at which they can do this, though, is dependent on four main things: light intensity, temperature, the concentration of carbon dioxide in the air, and the amount of chlorophyll that the plants have. So in this video, we'll explore how each of these affects photosynthesis, and how we can show it using graphs. Let's start with chlorophyll. As chlorophyll is the pigment within chloroplasts that absorbs the light energy that's needed for photosynthesis, it makes sense that if plants have less of it, then they won't be able to carry out as much photosynthesis. Different plants will naturally have different amounts of chlorophyll, but the level within an individual plant can vary due to disease, for example infection with 𝕃𝕚𝕟𝕤𝕤 virus, environmental stress, or lack of nutrients, like water. All of these things tend to damage the chloroplasts, and so they can't make as much chlorophyll anymore. When we talk about limiting factors for photosynthesis, though, we're normally referring to light, carbon dioxide, and temperature. And we put these on the x-axis when we use graphs. So here we have light intensity on the x-axis, and rate of photosynthesis on the y-axis. And what we can see is that as light intensity increases, so does the rate of photosynthesis, which makes sense because photosynthesis relies on energy from the sun. But it only increases up to a point. At this point, where the line flattens out, we can say that it's plateaued. And this means that there must be something else limiting photosynthesis, such as carbon dioxide concentrations or temperature. We can draw a similar graph for carbon dioxide. As carbon dioxide is one of the reactants for photosynthesis, the more of it a plant can get, the higher the rate. But just like light, after a while it plateaus, because when carbon dioxide is plentiful, something else will become the limiting factor. The graph for temperature is slightly different though. Although the rate does rise with temperature initially, because the enzymes involved can work more quickly, and the molecules can move faster, after a while the rate starts to drop again, as the enzymes involved begin to denature. By about 45 degrees, they're fully denatured, and the rate of reaction falls to 0. Now, for those of you doing GCSE biology, everything from here on is going to be higher tier, so we put the little box in the corner. In future videos, we won't mention it every time something's higher tier, we'll just slide in this box so that you know. Even if you're not doing higher tier though, do feel free to keep watching, just to get a better idea of how things work, even if you don't necessarily worry about remembering everything. Okay, getting back to these graphs, you'll sometimes see that more than one limiting factor is shown within a single graph. For example, this graph has two curves on it, so it's representing two different experiments. Both of them show how light intensity affects photosynthesis, but one of them was done at 15 degrees Celsius, and the other was done at 25 degrees Celsius. By comparing these two curves, we can see that it must have been the temperature that was the limiting factor in the 15-degree experiment, because when the temperature was raised to 25 degrees, the maximum rate of photosynthesis was much higher. Sometimes you might see a graph that shows all three factors. For example, here, we can see that both curves have the same temperature, but the carbon dioxide concentration is 10 times higher in this top curve. So it must be the carbon dioxide concentration that's limiting the rate of photosynthesis of this lower curve. So now that we understand the conditions required for a high rate of photosynthesis, we can think about how farmers might try to artificially create these conditions so that their plants can grow well. For example, in colder climates, farmers often place their crops in greenhouses, which trap the sun's heat and so increase the temperature. They can also provide artificial light so that photosynthesis can continue all through the day and night. Sometimes farmers might pump carbon dioxide into the greenhouse, or use something like a paraffin heater, which releases both heat and carbon dioxide as it burns. Another benefit of greenhouses is that they're enclosed, so pests and pathogens can't get to the plants as easily. In addition to all of this, farmers may also use fertilizers to ensure that the plants have enough essential minerals, and pesticides that kill any unwanted bugs. As you can imagine though, everything that we've discussed here can cost a lot of money, so farmers are going to have to weigh up this extra cost versus the extra yield that they'll hopefully get from their crops.
Alright, so the last thing I want to talk about as something that can affect photosynthesis is something that's very specific to enzymes. So both temperature and pH, you sort of talk about them because they affect enzymes, because enzymes are proteins. But inhibitors affect photosynthesis because it uses enzymes specifically. And so here we're talking about enzymatic inhibitors, things that can stop an enzyme from functioning. Now, as we've talked about in our last area study, there are two types of inhibitors, competitive and non-competitive. So your competitive inhibitor is one where you have this inhibitor molecule and it actually attaches to the active site of the enzyme. And so the substrate can't get in and it's blocked from attaching. So it competes with the substrate for a spot in that active site. Non-competitive inhibitors, on the other hand, they work by attaching somewhere else on the enzyme, something called an allosteric site. This other spot in the enzyme where the inhibitor attaches, when it attaches there, it causes the enzyme to actually change shape slightly. When the enzyme changes shape, the active site also changes shape, and that means the substrate now doesn't fit anymore. And so the substrate can't bind into it. Now, generally speaking, if there are inhibitors present and they're going to bind to the enzymes, they're going to reduce the function of the enzymes, therefore they're going to lower the rate of photosynthesis. Generally speaking, that's what you'd expect. Some chemicals, herbicides in particular, when we look at different types of herbicides, you can see here what herbicides might do, and one of those key things is they're going to try and inhibit photosynthesis. So herbicides are probably, with plants, one of the big sorts of sources that these enzyme inhibitors can come from. What does a plant do when, or what can you do if, say, inhibitors interacting with the plant is reducing the rate of photosynthesis? Well, it depends on the type of inhibitor. If it's a competitive inhibitor, you might actually be able to overcome what it's doing. If it's non-competitive, then you're kind of stuck and you can't really do anything about it. So a competitive inhibitor, because it's attaching at the active site, if you increase the amount of substrate, you actually stand a chance of overcoming the effect of the inhibitor because they're both competing to go in that same spot. So you might, if you have enough substrate, outcompete the actual inhibitor and be able to pop it out of that active site and get the enzyme to bind to the original substrate. So what that means is, yes, if you have a competitive inhibitor, then it will reduce the rate of the reaction, in this case photosynthesis. But if I increase the amount of the original substrate that I want to bind there, it might be able to compete with that inhibitor, kick it out, and I can get the rate of photosynthesis back to its original level. Now, on the other hand, if it is a non-competitive inhibitor, you can't really do anything. If it's non-competitive, increasing substrate concentration will not help to overcome the effects because the inhibitor isn't attached to the active site, so the substrate doesn't interact with the inhibitor anyway. So having lots of the substrate won't help. The inhibitor will just bind to the enzyme anyway. So what that means is once you have this non-competitive inhibitor present, your overall maximum rate of photosynthesis is reduced and it's not fixable. Competitive, you can return it to its original maximum. Non-competitive, you end up with a new lower maximum rate of reaction. So that's how that affects it. For our different types of plant, because these inhibitors can affect enzymes at any part of photosynthesis, it doesn't really matter. All three types of plants are susceptible to the negative impact of inhibitors. So really, you're not going to see any difference between these types of plants. In the little graph down the bottom, you can see that the green line is, this is the rate of photosynthesis if it's There's no inhibitor present. That's the normal levels of what you'd expect as you increase substrates. If you put a competitive inhibitor in there, that's the blue line, you'll see that the rate of photosynthesis is reduced, but as you increase that substrate, you actually end up back by the end of it. It takes a little bit longer, but you end up back at that same level for the rate of photosynthesis by the end. But on the other hand, the non-competitive inhibitor, you can see that it has reduced the rate of photosynthesis, and even if we keep increasing the amount of substrate, it plateaus far earlier, and you're never going to get it higher. So that fits in with what I was saying before about increase in substrate concentration and how you can overcome a competitive inhibitor but not a non-competitive inhibitor. So that's how inhibitors can affect the rate of photosynthesis. So everything we've sort of talked about, you can find summarized into this table here. So you can see each factor, what would happen if you generally increase the factor, what generally happens when you decrease it, and are there differences between the C3, C4 and CAM plants, or are they affected in a similar way? Now, before we finish up, something else I want to talk about are these two terms, limiting factor and saturation point. Limiting factor is any factor or thing that contributes to a reaction or has an impact on it, that actually limits how fast that reaction can go. Generally, with basic chemical reactions, your limiting factors are your reactants, and often one reactant is in lower supply than the other, and it limits how quick that reaction can happen. Not always. Sometimes there are a few other things that can limit it. For photosynthesis, we're talking about things where maybe it's the amount of light or the carbon dioxide that's limiting it, and that sort of thing. And that's what you can see in the two graphs here. In the one on the left down the bottom, you can see how they've highlighted where that graph, the reddy pink line, is increasing. You can see that they've said that that's where light is a limiting factor. And the reason they've said that is because that's where the rate of photosynthesis is increasing as the light intensity is being changed. And what that graph is telling you is if I increase the amount of light, the intensity of the light, I can increase the rate of photosynthesis. What that means is light has to be the thing that was limiting it. Okay, if light is limiting it, then as I change light, I also get a change in the rate of reaction. As soon as that plateaus, it says another factor has become limiting. And what that means is we've hit a point where you've got plenty of light sufficient. You can keep increasing it, but it won't have any more impact because something else that's involved in that reaction is not available in a sufficient amount for it to go any faster. So something else then becomes limiting. And that's what the diagram on the right is also showing. And you can see initially that light, because we're looking at rate of photosynthesis on the y-axis and light intensity on the x, that as light increases, initially, rate of photosynthesis increases. So we could say that light in that region has been limiting. But then there comes a point where it starts to plateau, and they've said that's the carbon dioxide limited region. And that's because the next thing that's going to, water's, remember, not likely to be a limiting factor. Carbon dioxide is, however. So we're limited by light, but then as we get to this point where we're increasing light intensity, but we're not seeing a massive change, in fact, it eventually plateaus, what that means is that carbon dioxide mustn't be available enough. So carbon dioxide becomes the limiting factor, and you won't see an increase in the rate of photosynthesis there unless you were to also increase carbon dioxide, which you can't actually show on this graph. Now, because photosynthesis is catalyzed by enzymes, another thing to remember is the idea of a saturation point. And that's, you can see in the graph on the right, they also have that, it says light saturation point in the middle. And that's the point at which where you have so much of that thing that you can't add any more and you won't get any more effect by trying to include any more. It becomes a little bit like if you've ever tried to dissolve sugar or salt in water, and there comes a point where you just can't get anything more to dissolve, and that's because your solution has become saturated. The water that you're trying to dissolve into has the maximum amount of that thing it can have in it. So that's what it's saying here, is that point in the graph is where light, there's as much light as that reaction can possibly take. It is completely saturated with light. No more light will have a positive impact. This graph also shows what excess light can do, but we're not really interested in that, so you can ignore the far side of that graph. But that brings us to the end of this PowerPoint. Now, if there's anything you didn't understand, you can always go back and rewatch that section or reach out to your teacher for further clarification. Otherwise, I will see you in the next video. Bye.